Dear student, today we are going to talk about a concept in genetics called genetic linkage. You already know that genes are found on chromosomes, and chromosomes are like long string of information inside our cells. So now when we talk about genes, we can think of them being passed from parent to their offspring or children. Sometime what happens? Genes which are located close to each other on the same chromosome can get passed down together. They don't separate as much as we might expect. And this is what we call genetics, genetic linkage. Suppose you can think like this. Suppose two friends are holding hands walking down a street. If they are holding hand tightly, and it is much harder for them to go in different directions. Similarly, if two genes are very close together on a chromosome, it is harder for them to separate when chromosomes are suffered during the process of making eggs or sperm. But if genes are further apart on the chromosomes, they have a better chance of being separated, just like friends who are not holding hands might walk away from each other very easily. So, in simple terms, genetic linkage means that genes are physically close to each other on the same chromosome, tend to be inherited together, and this is really very important for understanding how certain traits that run in a family or families are passed on from one generation to next. So in this lecture we will discuss this concept in subsequent parts, but for now you have to remember that genetic linkage is like two genes being linked together, and they like to stick together when passed down to the next generation. So, scientifically, we define the linkage like this: the tendency of two or more genes which stay together during inheritance or during gametogenesis is known as linkage. And linkage is the consequences of the concerned gene being together in the same chromosome. And linked genes do not show any segregations, due to which the ratio we generally obtain in F2 or in backcross are significantly different from expected ratios. In case of F2, if independent assortment are there, genes are segregated independently, then we will get 9 is to 3 is to 3 is to 1 ratio like this. This is the expected ratio we will get. And in backcross, this ratio is 1 is to 1 is to 1 is to 1. So, if we take an example, let us take the example say this. Now, the way the genes are arranged in parents can affect the kind of gene combination we might see in their children. So, in this diagram, you will see One parent has both A and B versions of gene, which are called dominant gene. These are A version and these are B version of genes. So their genotype will be A, capital A, capital B, capital B. If we talk about other parent, it has both A version and B version of genes, which are called recessive alleles. So their genotype will be AABB. Clear? When these two parents have children, now when the crossing taken place, then what kind of children they will have? So when these two parents have children, they pass on one gene from each pair. So this is one pair, this is one pair of other parent. They pass on one gene from each pair. In this case, capital A and capital B are linked together in one parent, and small a and small b are linked together in other parent. The children will mostly inherit these pairs, right? This means the children will mostly look like their parents. But it might be possible, it might be possible that during reproduction, these genes can shuffle also or mix up, which creates the new combinations, which is called recombination. The new combinations are, you can say, these new recombinations, you can say, they are recombinant type. Clear? So, in your first diagram, what we get? When A pairs with small b, when this capital A pairs with this small b, a new recombinant type will form. When B, or you can say, or it's vice versa. So you will get in this case, which is known as coupling phase, four types of gametes we will get. First one is capital A, capital B, which is of course parental type because A and B they are paired together. Small a, small b, again we have parental type like this. And due to shuffling, we will get two types of genotypes, capital A, small b, and small a, capital B. These two types are recombinant types. Then there is another phase which is known as repulsive phase. In repulsive phase, what happens? In repulsive phase, one parent has genotype say capital A, small b, which means they have A version of gene and B version of other. In second parent has, or you can say second parent has the opposite genotype, say small a, capital B, meaning they have A version of one gene and capital B version of other. In this case, the dominant allele A linked to the recessive allele B and recessive allele A linked to the dominant allele B of other parent. When these parents have children, they pass on these combination. So children may inherit mixed version of genes. Like in first diagram, you will see sometime recombination may happen and genes can suffer. This, mean, this means that children could end up with new combination like capital A, capital B, or it may be small a, small b, which are different from the usual combination their parent have. So in this case, in repulsive phase, we will get just opposite of the coupling phase. How? Let us see. So in second diagram, the gamete produced are, again we will get four types of gamete. First one is capital A B, which is same as that of parental type. So it is parental type. AB like this, again parental type. But we will get capital A, capital B, which is not same as that of their parents. So it is recombinant type. And small a, small b, again it is a recombinant type. So in coupling phase and repulsive phase, the gamete produced, their type is just opposite. Right? Now, in coupling phase, the recombination type means new allele combinations are capital A small b like this and small a capital B. But these are parental type in repulsive phase. Here you will say they are parental type, capital A small b and small a capital B. So in repulsive phase, the recombination types are capital A, capital B, and small a, small b. But they are parental type in coupling phase. So you can think coupling and repulsion phase as opposite of each other. So in the coupling phase, capital A and capital B are on the same chromosomes like this, and small a, small b are on the other chromosome. So the parental types are one where allele from the same parent inherited together. It means these two genes are inherited together and these two genes are inherited together. So parental type are ones where allele from the same parents are inherited together. Capital A, capital B or a, small b. And recombinant type are new combination created due to crossing over or recombination. So you will get capital A small b or small a capital B. And as I have already told that in repulsive phase, A is paired with small b and small a is paired with capital B on different chromosomes. So in this phase, the parental types are ones where dominant allele of one gene is paired with the recessive allele of other gene. So we will get capital A small b or small a capital B. The recombinant types are those where allele are rearranged from their parents' chromosomes, say capital A, capital B, and small b. So, in nutshell, you can say coupling and repulsive phase they are opposite of each other. Now, we will talk about what type of linkage we are going to study in this lecture. So, type of linkage. So, we will talk about the linkage in genetics, and linkage is about how genes are inherited together. These are of two main types. First one is complete linkage. In complete linkage, we only get the parental type of offspring. This means that the genes are very close together on the chromosome and are always inherited together. But in case of incomplete linkage, we see both parental types and recombinant types. Recombinant, as I have already told, that are the new combination of gene that come from crossing over. So both parental and recombinant type we will get in incomplete linkage, and so the mixing of gene may occur due to which the recombinant type can be produced. Now let us discuss what happened during the crossing over. So what is crossing over? So crossing over is nothing but it is exchange of genetic material during meiosis, and it generally occurs in recombinant types. So if we want to discuss what happened during crossing over, when crossing over occurs, we usually find only recombinant type. However, in general, there is a partial linkage. This means that some gametes, which are the cell that will become egg or sperm, are more common in other. So, in case of partial linkage, some gametes are more common than other, and now we tell about something about independent assortment. What is independent assortment? Means if independent assortment is there, then 50% parental type will be produced and 50% recombinant type will be produced. If such is the scenario, then we can say genes assort independently. And if parental type is not equal to the recombinant type, you this may also happen. So when parental type is not equal to the recombinant type, it suggests that there might be linkage, but we can't be completely sure because other factor might also affect the result. So when parental type is not equal to the recombinant type, then it is possible that the linkage may be there, but we cannot confirm due to various factors, right? So let us remember that we are often looking at a single locus with two allele. An allele, I am again repeating it, is a version of a gene, and locus is the specific location of a gene on a chromosome. Now, we will devise certain mechanism by which you can detect the linkage. So, first of all, I just memorize you the Mendel's law of segregation and uh, how to test if what we observe matching the theory. So we know that Mendel's law tells us that in F2 generation, when you cross a dominant allele, say capital A, with a recessive allele, small a, the phenotypic ratio is expected to be 3 is to 1. I mean to say, if I consider this cross, so I will get three types of genotypes. In this case, I will get this when it is again crossed with this. So three types of genotypes are produced: capital A, capital A, capital A, small a, and the ratio is 1 is to 2 is to 1. Now, phenotypically, these two classes are same. So phenotypically, their ratio is 3 is to 1, right? So you will expect a ratio of 3 is to 1. This means three parts dominant, means capital A, capital A, capital A, capital A, or capital A, and one part recessive looking, that is small a, right? But here is our key question. What if we actually observe some data in an experiment? How do we know if what we observed matches this theoretical ratio. When we conduct an experiment, we will get certain data. Whether this data matches the theory, for this we need some statistics. And that's where the chi-square test comes into picture. So suppose we have two categories. First category is dominant type. For example, this category is dominant type and other category is recessive type. So the dominant type is generally represented A dash and recessive type we represent it say like AA. Clear? So let us start. So first of all, in this diagram, this is nothing new in this diagram, means capital A is crossed with small a. So the gamete produced are A and A. So in F1, you will get this type of genotype. When selfing take place, means F1 is crossed with F1. In F2, you will get three types of genotype. Out of these three, two are of same type, dominant type, and one is of recessive type. And their ratio is 1 is to 2 is to 1. And their phenotypic ratio becomes 3 is to 1. Right? Then, if I am saying same thing, if capital A is dominant over small a, then phenotypic ratio in F2 is 3 is to 1. This is theoretical ratio. Now, given for F2 generation, then is the observed ratio is agreement with theory. For example, if you conduct an experiment and in which you get a ratio and you want to confirm this ratio is in agreement with the theory. So to answer this, in case of two categories, we can use Z test for testing the significance of given proportion. But generally we will use chi-square test. So how this chi-square come into picture? So chi-square is given as sum of observed minus expected whole square divided by expected. Suppose we have k groups. So I is equal to 1 to k. Then the degree of freedom is k minus 1. So now we will assume that the sum of observed values is equal to sum of expected and it is further equal to n, means total number of individuals. Now consider the general case. For this condition, the phenotypic ratio is 3 is to 1. We want to test our observed ratio is in agreement with 3 is to 1 or not. What we do? Suppose we have got two categories. First one is say dominant phenotype, as I have already told. That include and capital A capital A small a. And recessive phenotype small a. Let us say we counted the observed number in these categories. Suppose in our experiment, A1 number of individuals belongs to dominant type and A2 individual belongs to say recessive phenotype. Then in order to test whether the linkage is there or not, we will use chi-square. So in this case, we have two categories. So the degrees of freedom for chi-square is 1 degree of freedom, 2 minus 1. So you will compute the chi-square with 1 degree of freedom as A1 minus 3 by 4 n square divided by 3 by 4n plus a2 minus 1 by 4n divided by 1 by 4n. Now question arises from where this 3 by 4 comes in. Actually these are the proportion. So in order to get the expected number, what we do is we multiply this ratio with the total number. So out of 4, there are 4 numbers, and we are assuming that there are n individual. So 3 by 4 n is the number of individual belonging to this category, and 1 by 4 n is the number of individual belonging to this category. So I have the expected, and I have say observed. Observed is my A1 minus this expected, its square divided by expected. Similarly, for second class, this is my observed minus 1 by 4 is expected number divided by 1 by 4n. Now I want to simplify this. How this can be happened? So if I simplify this, what I will get from this 3n is the denominator. This 4 goes here, and I expand this square. So a1 square plus 3 3 is 9 by 16 n square minus 3na by 2. Actually, it is 6n a1 by 4. So this 3 cancelled with 2. So it is 3na by 2. Similarly, for this 4 goes here. This is a2 square plus n square by 16. 1 by 4 ka 1 by 4 square, it is 1 by 16. N square, it is n square minus, it is again 2a2 n by 4. So divide it by, so it is n a2 by 2. Divide it by n. Now I simplify. If I take the LCM of these two, then what happen? So it is 3n. So first term as it is 4a1 square plus 9 by 4. This 4 cancelled with this 4. So it is 9 by 4 n square. Again, this 2 is there and 4. So 2 multiplied by it is 3 n a1. Now this 3 goes here. So it is 12 a2 square plus 3 by 4 n square minus 6 n a2. This is very simple mathematics. Everybody can do it. So if we further simplify it, what we will get? We will get like this. This 4a1 square plus 9 by 4 n square minus 6 na as such plus 12 a2 square plus 3 by 4 n square and 6n. This a2 can be replaced here because we know that the number of individuals in any cross we will get n. So a2 will be equal to n minus a1. So I just substitute here and simplify it so that something may be cancelled. So in this case, first part as such, second part as such. Now I open it. This is minus 6n square plus 6n a1. Now this cancelled out. And further simplify it, it will become 4a1 square plus, I forgotten here, actually it is 9 by 4 n square plus 12 a2 square minus 3 n square. Now from where this 3 comes? This is 9 by 4 plus 3 by 4 n square. So 9 by 4 plus 3 by 4. So it is 12 by 4. So it becomes 3. That's why I ignore it. So minus 3n square is there. You just simplify these two terms and you will get this n square divided by 3n. Now I take 1 by 3n, then 4a1 square plus 12a2 square minus 3 times n square is again replaced with a1 and a2. Because n is equal to a1 plus a2, so I substitute a1 plus a2 in place of n square. So now further simplify it. It is 1 by 3n 4a1 square plus 12a2 square minus 3a1 square minus 3a2 square minus, when I open this, 6a1a2. So you will get 1 by 3n 4a1 square minus 3a1 square. So it is a1 square. 12a2 square minus 3a2 square. It is 9 minus this 6a1a2. And chi square with 1 degrees of freedom can be obtained. Say it is nothing but a square of a1 minus 3a2 whole square divided by 3n. So thank you very much.